Good afternoon, everyone. Welcome to the fourth webinar on grassland restoration, organized by the EXPBIO-Project. I'm Katrien van den Bussen from the Vlaamse Landmaatschappij, the Flemish Land Agency, where I work in the Department of Agro-Environmental Schemes. EXPBIO is an EU Interreg North Sea Programme project and is a collaboration of 8 partners across Sweden, Denmark, Germany, the Netherlands and Belgium. We are municipalities, universities, botanical gardens, public research institutes and governmental agencies. The EXPBIO project is focusing on restoration of grassland biodiversity and bringing back flower-rich habitats in the North Sea region. By combining expertise from different countries, we are exploring practical management practices and creating tools to help the local plant communities augment. Many of you have already listened to the previous webinars. We started this spring with a webinar series on seed mixtures and seed cultivation. Today, we continue this journey with 6 inspiring speakers sharing new insights and proven strategies for restoration and rediversification of grasslands across Northwest Europe. In the previous 4 webinars, we learned about different management methods for obtaining species-rich grasslands, from mowing methods to fire and herbivory to sowing. Sometimes it isn't sufficient to restore plant communities and we have to translocate species. So I'm very grateful that Sandrine Godefroid is here today to explain us why and when translocation of rare plant species is recommended. She will show us the techniques for implementing this type of action and the points of attention. Sandrine Godefroid works as a scientist at the seed bank of Meise Botanic Garden. Since 2009, she has been collecting seeds of rare species in Belgium for conservation and scientific purposes. Among other things, she studies their germination, ecology, and storage behavior, and she is also involved in seed-based restoration of degraded ecosystems in Belgium. The webinar will last approximately 1 hour, with 30 to 40 minutes for the presentation, and afterwards we will have time for discussing. Some technical issues. Gunilla Hell-Bellman, you see her on the screen. She's in charge of the technical aspects. She will ensure that everything runs smoothly. We ask you to mute your microphone and to use the chat for communication and for the questions. With that, I give the floor to you, Sandrine.
Thank you, Catherine, for the introduction. Thank you also for the invitation. Hello, everyone. So today I'm going to talk about how the translocation of rare plant species can complement grassland restoration. And I'm insisting on the word rare plant species, because for common species, you can consider other techniques like sowing, things that have been frequently illustrated in the previous webinars. But here it's really about rare plant species. So let's first define what is a plant translocation. So if we take the IUCN definition, the first point is that conservation translocation is the deliberate movement of organism from one site for release in another. The second important aspect is that it must be intended to yield a measurable conservation benefit at the levels of a population, species, or ecosystem, and not only provide benefit to translocated individuals. So this is very important point. And conservation translocations consist of reinforcement and reintroduction within a species indigenous range. So if it's outside the species range, it's not a conservation translocation, it's an introduction, and it's something that is totally different. So we are talking about reintroducing things within their indigenous range. So let's see why translocations is sometimes necessary. Well, it's important to keep in mind that most target species will not spontaneously come back to areas where they were present. And this is due to several reasons. The first one is the short-lived soil seed bank. If we look at the database of Thomson quite some time ago about soil seed bank types of Northwestern European flora, you can see on this graph that actually barely 17% of the Western European flora has seeds that remain viable in the soil for more than 5 years. So that's what we call long-term persistent soil seed bank. The second aspect is that many species have a limited dispersal capacity. If I take this graph from the study of Lososova and colleagues, having a great sample size of more than 10,000 species from the European flora, we see that actually a quarter of the European flora has a natural dispersal distance up to 1 meter for 99% of their diaspores. 57% can disperse up to 5 meters, and 71% can disperse up to 15 meters. So these are these bars in the histogram. So actually, most European species have unassisted dispersal, which is limited to a few meters. So that's a second issue. And then we have to keep in mind that many species will not persist in the long term if their populations do not have a minimum viable size. So what is a minimum viable size? Well, there is no magic number, actually. There is no size that fits all the plant species. So we don't know how much it is for most of the species. But what we do know for sure is that there is a positive correlation between population size and several variables that measure the plant fitness. Like in this example of Gentianella germanica, you have the number of seeds per plants against this population size. So you have a nice correlation here. It's the same for the number of seeds per fruit, for Primula vulgaris, for instance. If we look at another fitness metric like offspring size, you get the same kind of relationship. And then I put here a meta-analysis from some time ago, where the authors have looked at the correlations between fitness parameters and population size. And for 45 studies, they have found a mean correlation of 0.4. So you have that kind of correlation for several species and probably many more than that. So considering these three aspects, like the soil seed bank, the dispersal capacity, and the viable population size, we can actually consider that these species having this kind of problem may need to be translocated. So I mean, increasing the size of the populations that are still there, the relict populations, that's what we call a reinforcement, or for those populations that have been extinct, so to restore these extinct populations. That's what we call a reintroduction. And both terms are in the overarching term of translocation. The fact is that species translocation is increasingly recognized in international treaties like the Berne Convention, the CBD, the Global Strategy for Plant Conservation, and so on. And it is actually So how can I cannot. My slide is frozen. Yeah, okay. Sorry. It is actually an increasingly used conservation approach worldwide. If you look on this small graph here, where I have put the number of peer-reviewed papers on plant reintroductions against the years, you see that there is a huge increase. And if we look at some reviews that have been done at a global or at a national level, or even at a regional level, you can see that a lot of plant taxa have been translocated in a number of, a huge number of different populations. So now that we know more or less why should we consider sometimes to translocate a rare species, let's see how to do that. So first of all, you need a material. So if the species is locally or globally extinct in the wilds, then you need to consider using ex situ stored seeds. So as you probably know, there are all over the world different ex situ seed banks that are conserving seeds in proper conditions with climate chambers that are drying the seeds down to 5% moisture content and conserving them at -20 degrees. So this ensures the conservation of seeds for decades. If the species still exists in the project area, then it's always preferable to use fresh material. For the extremely rare species, you have no other choice but collecting manually because the species is too rare in the landscape. If your species is rare but locally abundant, you may consider collecting using machines because this probably is much efficient. So how to choose the seed source populations? Well, it's always preferable to use stable populations as seed sources compared to decreasing ones, as you can see here from this graph, which is a review of translocations globally. You can see that if we take, for instance, the survival rate of these populations, after 1 year monitoring or 3 years, well, we can see that especially after 3 years, you can have a much higher survival rate in these populations when seeds are sourced in stable populations. So the reason is that declining or small populations can lead to loss of genetic diversity. They also can have accumulation of deleterious mutations. They also can have increased inbreeding and inbreeding depression. So now that we have the material, we need to propagate it. As we are working with rare species, we have a limited number of seeds, and so we need to propagate it because we cannot just sow 3 grams of seeds. So we're not going to restore anything with that just by sowing. So let's propagate it to have more plants. You can germinate the seeds in the lab in controlled conditions. So this ensures a higher germination rate and a less spread out germination time. But you can also use for germinating the seed directly in the greenhouse in potting soil, which is also good. And the advantage is that it prevents the seedlings from being transferred from the agar, which is the medium in the lab, to the potting soil. And this is a very delicate stage. So it can be advantageous also to seed in the greenhouse directly on the potting soil. It's important to keep in mind that in this framework, propagation time must be limited to a few months. Otherwise, we may have an increase in the frequency of plant threats that are typical of a production environment, but poorly adapted in the wild. For instance, a loss of drought tolerance because the gardeners are regularly watery. And we can also have a loss of competitive capacity because the gardeners are regularly weeding. And so this is obviously not what we want in the frame of a translocation on the field. And so I put here some photos of species we have been growing. And so this is how they look like just before the transplantation on the field. So let's see now the habitat restoration. So to implement a translocation, you need a habitat. And if the habitat to be restored has been completely destroyed, it happens sometimes, well, you need to prepare the conditions to receive the plants. In this case, I've put some photos on a case where in the frame of a live project, plantation of pine trees has been cut down. Then you need to clean, of course, the area, piling the branches, removing wood debris, sometimes considering sod cutting, especially in the Belgian context where you have, in some cases, you can have a very high phosphorus level in the topsoil. Then you may consider implementing sod cutting as well. And to transplant your focal species, I would recommend using at least 500 plants, just to try to reach this minimum viable population size. So again, we don't know how much we should reach for each target species. It may be 500, it may be 300, but for sure we are talking about hundreds of individuals, just to avoid having some genetical problem afterwards. Also knowing that we will have some mortality on the field. And actually, if you look at this graph, we see that there is some kind of correlation between the number of reintroduced individuals and the survival rate, especially after 1 year and 3 year monitoring. So there is some kind of nice correlation. And if I take the results from another study from Bellis and colleagues, who made a review of translocations in the US, well, they say basically the same message. They have found that what they call the founder size, so the number of translocated individual, is the best metric that explain translocation success. So large founder sizes increase population persistence, which is the survival rate, and recruitment into the next generation. Let's go now to the origins of the seeds, and especially the fact that we recommend mixing several origins. This is to increase the genetic diversity and therefore the resilience to future stochastic events. So I've put here some image, which is an extract of a plantation design. And you see that, every plant, every individual has a code, starting with a letter and then a number. And the letter refers to the origin of the seed. And so you can see here that we planted next to each other plants having different origins. And why did we do that? Well, because of the pollinators and because of their behavior. They are a bit lazy like humans. So if they find a plant that is suitable for them, very close to them, they will fly and go and pollinate this individual that is close. And they won't make the effort to fly 10 meters away to find exactly the same species if there is another individual closer by. So we did this for trying to maximize the gene exchange between individuals of different origins. And actually it works. According to our genetical analysis, we have a good mixture between the genotypes in the recruits. So now that the plants are on the field, we need to monitor them. And the variables used to assess translocation success among monitor projects are more or less these ones. At least this is the results that we had in a recent review of plant translocations in Europe. And you see that those variables that are most frequently used for the monitoring of these translocated populations are survival, reproductive factors, and the recruitment. And I'm not going into the details here because monitoring in itself is quite a complex matter, but I invite you to have a look at the species recovery manual that has been published a few years ago by BGCI, so Botanic Gardens Conservation International. And so we have written a chapter on the monitoring where you can find all the details, what to monitor, how, when, and so on. And so we have divided this chapter into the demographic monitoring, the genetic monitoring, and the ecological monitoring. So for further details, please have a look at this publication. So now I'm going to briefly show you some examples of projects where we restore grasslands in Belgium. And one of the approaches to restore these grasslands is plant translocations. So the first project was the live herbages. It is a project that has been completed 6 years ago. So we restored a little bit more than 600 hectares. We were working on these habitats. There were 3 partners, 1 NGO, 1 environment agency, and my institution, so Meise Botanic Garden. So it was a live project, so partly paid by the live program. And for these kind of projects, so the actions are, well, frequently the same. So first, land purchase, because you need to keep control on the land you are restoring, especially towards the future. So this is done by the NGO, an environmental agency, as well as the habitat restoration. So you first need to restore the right conditions to translocate the species. And this can be, well, quite different depending on your situation. So we have seen previously some pictures on cutting exotic trees, but it can also be pulling out just brambles, removing topsoil, also seeding the structure, the species that are going to structure the grassland. So the characteristic species that are quite common that you can collect mechanically, and you can seed hectares. Then fencing, just in order to avoid that you have degradation by wild boar or big herbivores. And then, yeah, grazing or even mowing, depending on the habitat types, by sheep, goats, donkeys, and so forth. So this is done by the partners, so the guys who are working on the field. And then we can, for some rare species, we can consider translocating the species as a complementary approach together with seeding other more common species. So these are the 4 species we were working on, together with their conservation status in the target area. So we were introducing in total 17 populations of between 500 and 700 plants each. So in total, it means a bit more than 9,000 plants. So a few pictures about the process from the nursery to the field, the day of the plantation. Then 2 years later, you see that many species are, many individuals are flowering. So this is the example of Arnica montana. You see at the beginning, you see a line pattern because we plant it in line because this facilitates the monitoring. But you see that after a few years, so a few years later, this line pattern has disappeared. Obviously, because you have recruitment, so you have many more individuals, and you have also some mortality among the transplants. And so here you can see that a lot of new rosettes have appeared 6 years after the plantation. Another example of a similar project, again in Belgium, is the Life Connection project, which is still ongoing. So we are working on 6 different grasslands and 2 different forest types. It's again a live project, and we have basically the same kind of actions. So here we are translocating 14 different species, typical of habitat 6210, which is calcareous grassland. So 5 species were translocated as plug plants. So actually 4 of these were already translocation, and we are still working on the last one. And 9 species have to be sown because either they are annuals or orchids or species with very tiny seeds that are not easily to propagate. So we can then use plots where we sow these seeds on a very small scale. And for orchids, it works pretty good. So then a few images again. When the seeds are sown in the lab, then after transferring the seedlings in the multi-trays after 6 weeks for this Pulsatilla vulgaris species. And then this is how the plants look like just the day before their translocation on the field. A few photos the day of the transplantation. So, and the year after the translocation, so a lot of individuals are starting to flower. And the last example I wanted to show you briefly, again, a live project in Belgium, still ongoing. We are working on many different habitats. Here there are 7 partners, 2 NGOs, 2 universities, 1 environmental agency, 2 foundations, and my institution. And so the actions are basically the same as the 2 other projects. Here we are translocating 20 typical species of these 5 habitats. So we plant 20,000 plants in total because we plant 2 populations per species of 500 plants each. So these are just a few examples of the species we are working on, again with their conservation status. And one is already extinct in the wild. So we have to rely on the ex situ stored seeds. Here a few images for Gentiana pneumonante, which is 6 months old on this image, just before the transplantation on the field. And again, a few pictures of the day of the transplantation. So let's look very briefly at some results. I cannot show all of them, but so let's highlight some of them. And especially the relationship between the size of source population and the quality of the material used. So we have a kind of a positive correlation between the flowering population size and the genetic diversity, at least for Dianthus deltoides. Here the genetic diversity is expressed as multilocus genotype diversity or expected heterozygosity. But we have also the same positive correlation between population size and seed quality. And this is, for example, the results for Arnica montana. And seed quality can be expressed either in seed weight or germination rate or proportion of empty seeds. If we look at the effect of seed origin on transplant performance, so here in orange we have the results for Arnica montana, in bluish, so Campanula glomerata, and in greenish, Helichrysum arenarium. So we see that the seed origin influences the performances of translocated plants. So here we have the survival rate, or the rosette diameter, or the number of flowering stems and flower heads. And you can see that, so we have different seed origins, and so they have a different influence on, for instance, here the survival rate, or the fertility. So they definitely have an influence on that. Here we have the combined effects of translocation site and seed origin on plant performance. And so again, we have Arnica montana, Campanula, and Helichrysum here, and the seed origins, the translocation sites. And so if we look, well, here, for instance, at the survival rate, we can see that for the same performance variable, plants of the same origin can behave differently in different locations. So here D is different from the B and so on. But we also see that plants of different origins can behave differently within the same planting site. If we take just a single site here, we have the two different origins, and so we obviously have a different survival rate with these two different origins. And the effect is actually long-lasting, because these graphs show the last year of monitoring available. So depending on the species and the populations, this means that these are the results for year 4 up to year 8. So the effect is really long-lasting. So for the restoration strategy, we recommend using multiple seed sources across several reintroduction sites. Then I was interested in to what extent are plant translocation outcomes influenced by seasonal temperature and precipitation. So this is, well, work in progress, but the very first and very simple analysis have shown that there is probably something to explore more. For Arnica montana, for instance, if we look at the survival rate, so we have the two different origins, and we have seen that obviously the mean temperature of the first quarter, the second quarter, the minimum temperature of the third quarter, they do have an influence on the survival of the Arnica montana. Also, there is something to say about the relative humidity of the first and the third quarter. And for the origin, we have basically the same results. And if we take another species, the Campanula glomerata, and if we look at the precipitation of the second quarter, and here we have the number of floral stems per plant, obviously this species prefers when the spring is dry. And this is the same relationship for each of the 5 origins. But well, this is very preliminary results, so we need to do much more exploration of this, and currently more in-depth statistical analyses are underway in collaboration with the University of Grasse. So concerning the recruitment, so the new generation, so I'm gonna give you here a few figures. So let's take the 4 species we were working on in the frame of the first project, so the one that is completed since 6 years. So this is the project for which we have, of course, a lot of monitoring data. So the translocation year is here, the number of translocated individuals. And in 2017, we counted the number of new plants that have been produced by the transplants. So this was already very encouraging. And then in 2020, I made the effort of counting the recruits for Arnica montana, so we have a huge number. So this is very encouraging. So we can say that seedlings are produced. New seedlings also begin to colonize the surrounding area because actually the temporary fence that we installed for the first or the second year, we removed it. And so now these areas are grazed as they should be. And so thanks to these animals, the seeds are propagated further in the area. So this is excellent for the long distance dispersal of the seeds. So it means that a population dynamic is underway and that translocated individuals that will die in the future can be replaced by new plants so that we finally have a dynamic like this. So the transplants will decrease during the time because even if they are perennial plants, they will inevitably die in a few years. We don't know when, but they will not survive for decades probably. Then you have an increase in the new seedlings, and so the resultant curve should be like this. So you have a slight increase in your population through the years. So some take-home message. Well, we can say that translocation of plant species can be successful, provided that we collect seeds from healthy populations, so I mean large and not declining population. We need to choose sites that meet the needs of the target species, and this may sound like something that is trivial, but according to a review that we have published earlier this year, we have asked respondents, what is, according to you, the reason for failure in your own translocations? And we see that actually poor selection of sites or microhabitat is responsible for 11% of the failures in the European translocation. So it's not something that is, very easy to choose the right site for the translocation. We need to ensure that competing species are completely eradicated before transplanting, and we need to keep in mind that there is, for some species, a soil seed bank, because weeds do have a long-lasting soil seed bank, and so they will come back. So we have to make sure that we do not plant too early. And if we look at the graph here, we see that weeds and competition are cited for in 13% of the failures in the European translocation. We need to plant a large number of individuals from multiple seed sources if we want to improve the success rate. And here we can see that too few individuals has also been cited as a reason for failure. And we have to protect the restored population with a fence because you have sometimes big animals, herbivores like roe deer, wild boars that can destroy your restored population. And actually, animal disturbance has been cited here also as a reason for failure. And we have to ensure ongoing management. And so this can happen thanks to the collaboration of the field workers, so the NGOs, so the people who are really doing the job on the field. So another take-home message is that we have experienced that close collaboration between botanic gardens and field practitioners in restoration project has several advantages. First of all, it provides partners with the expertise, skills and facilities needed for ex situ conservation and plant propagation. So I'm thinking about seed banks, propagation greenhouses, spaces for common garden experiments. Secondly, this collaboration allows for a more ambitious project than purely small-scale species translocation, as we frequently see in Europe and in the world. So a habitat-based approach is much more important than a species-based approach. So this is very important to me and important to keep in mind. And this collaboration ensures also adequate site preparation before and optimum management after transplanting or sowing species. It significantly increases the chances of a successful plant community restoration. And it contributes also to member states' obligations to restore their habitats to a good conservation status. So we have to keep in mind that to claim that we have grassland in a good conservation status, we need to have a minimum number of characteristic species. And these species have to cover a minimum area. So if we don't have these conditions, we do not have a habitat in a good conservation status. And also this kind of collaboration allows restoration to be coupled with research to advance the science and practice of translocations. So if you want to know a bit more about that topic of translocating rare species, so I invite you to have a look at some papers that we have published from these live projects. Dealing with different aspects like soil seed bank, like designing protocols, like the contribution of genetics, and so on. And also I want to emphasize the publication of a decision-making tool for restoring lowland grasslands in Europe. That was also the result of our experience in 2 different live projects. And this is actually a key to guide managers in their restoration approach. And by lowland grassland, here we mean these different grassland. And this decision-making tool is dealing with important aspects like the status of the target populations, the need for translocations, because it's not always needed, the soil nutrient richness, threat mitigation, about how origin of plant material, also how to propagate the target species, how to prepare the target site, the sowing period, site management in the years following the intervention, etc., etc. So these are the institutions we are working with in the framework of these translocation projects. And that was my last slide. So I would like to thank you for your attention, and I'm looking forward to answer your questions.
Thank you very much, Sandrine, for your very interesting and clear presentation. So we can go to the questions. I have a first question. What do you see as the main challenge for the near future?
Yeah, I think there are several challenges. So if I start with the Belgian context, I would say that restoring the abiotic conditions, so preparing the area to receive the species, is something that can be challenging because we have to cope with a high nutrient deposit. And so in many cases, we have to implement sod cutting. And sod cutting is something that costs quite a lot. So it's, yeah, technically quite a challenge, especially on huge area, if we have to restore hundreds of hectares. And this can be implemented and financed, I think, only in the framework of life projects, because otherwise we cannot afford to do that. And if we don't remove topsoil, especially in the Belgian context where we have a lot of nutrients, then we will never get a species-rich grassland. So this is probably one challenge, but more in the context of Belgium. Then I would say another challenge that I see also is climate change, because we are trying to restore at a certain place grasslands as they were present in the past, like decades ago. But the problem is that the conditions might no longer be the same, and in the future they might also change. So if we want to restore, let's say, calcareous grassland at a certain place where the current habitat is still, well, the current climate is still okay, this climate might no longer be optimal in the coming decades. So, and that's a huge debate because we want to restore things that were present in the past. And so that's why, yeah, there is a huge debate around assisted migration. So should we restore these habitats in another place, considering that the climate will change, so maybe moving towards the north or things like that. But this is heavily debated, and this is, yes, probably, yeah, something else. But I see that as quite a challenge.
Thank you. I go to the questions from the audience. First question, is it better to source from a stable non-local population than from a decreasing local population, even if that means missing local adaptations?
Yeah, that's a good question, actually. So you need to see what is available. I would say, indeed, if you have only local populations that are depleted, well, there are different possibilities. Either you try to collect from all these small populations so that you can get, you can collect from a lot of individuals so that you can increase your genetic diversity by just merging different small populations. Or you can also consider going a little bit further and collecting from big populations. But you need to keep in mind that you can have, in some cases, outbreeding depression. And so you don't, yeah, you have to go not too far away. And you have to look at, do we have more or less the same ecological conditions in the source and the target site. So actually the ecological distance between source and target is, to me, more important than geographical distance. Because sometimes you have species that have ecotypes, and you have populations that are growing on acidic soil, other populations are growing on calcareous soil. You are not going to mix them. And so if you have the right populations growing a little bit further away than your local population, and which you suspect is ecologically appropriate for your target site, then you can consider collecting further away. That's also what we did in some circumstances.
What's your experience success rate with the translocation of rare species? I'm thinking about long term plus 10 years.
Yeah, and so that's a good point, and that's the reason why I presented only a few results from the first of our project, because for the other ones, well, it's obviously too soon to conclude anything. So for our very first project, we have now more than 10 years monitoring. This is becoming something that is quite robust to conclude something. But anyway, we have seen many translocation projects where populations were collapsing after a few years. And so the monitoring should last for at least, I would say, 10 years, preferably longer. Because especially we want to look at the functioning of the grassland. So is it really a dynamic population? Is it interacting with the insect communities? And so for getting that kind of results, we need, well, a longer period. And so that's why in the guidelines, you see that the monitoring should last for at least 10 years. Otherwise, yeah, it's too short. And the problem is that many restoration projects that are funded by, for instance, the LIFE program, so they are for a shorter period. And so you cannot conclude anything with that. So after 4 years, you can just say, okay, my population is still there, but it doesn't mean that it will be functioning, it will be dynamic, it will be still there in 10 years. So that's the problem. And we are continuing the monitoring on our own fund after these live projects. So the longer, the better.
Another question, do you normally remove all turf before planting the plants? And there is another question related to this. I don't, I saw it in the chat. How do you manage the existing soil seed bank of non-target species at the restoration site?
Okay. So removing the existing vegetation depends on how your site looks like. So in the case where your habitat is no longer there, then you have to get rid of everything, the shrubs, the trees, and things like that, because we want to restore grasslands. So then we are working on bare soil to sow the structuring species and plant the rare species. In some cases, the vegetation has been restored some time ago, and then we have we are starting to have a nice composition, vegetation composition. And in these cases, we are not removing the vegetation, so we are supplementing the vegetation composition with the translocated plants. So it depends a bit on how your site looks like. And to get rid of the soil seed bank of the unwanted species, well, you need to wait a bit. And so what we had at the beginning is that we had Genista coming back. How is it called? Rubus species, Betula seedlings, and things like that. And so we need to remove all these seedlings again and again. Because it's not solved in just a single management event. And then afterwards, you will see that the soil seed bank of these weeds are depleting. And so you can come then with your plants. So it's actually something that, you need to be patient. It's not like if you are going to restore a grassland in 3 years, especially in those degraded areas. So you need to wait. I have to admit that at the very beginning, I was a bit skeptical on some sites. I was thinking, oh, this is not gonna succeed. But after now, more than 10 years, I see that, it's actually on the right way to become a biodiversified grassland. So, it's something you need different approaches depending on the local circumstances, and you need to be patient.
Do you grow the plants in soil similar to the donor or the receiving habitat?
Actually, as we grow the plants just for a few months at most, they don't have the time to adapt to the nursery condition. And so it's not necessary to grow them on the local soil. We just use normal standard potting soil with some sand to make the mixture a little bit lighter. And it works for most of the target species, especially of dry grasslands. It works on standard potting soil. And so you don't need to take soils from the target areas. It's not necessary.
Someone asks to get a chart and literature advices for the things that are published.
Yes. Well, the person can contact me for further details. So the papers that I have put on one of the last slides are just a few of what we have published, and so well, we can get in touch with the person so that I can offer more information and publications, and if needed.
What have been your experience of sowing orchid species directly?
Yeah, actually, if your target site has not been completely restored, like moving lots of ground, disturbing the soil profoundly, normally the mycorrhizae should be still there. And so if you are just sowing on, well, bare soil or soil where you have made some gaps, then you can get some results. But you need to be a lot more patient than for the other species, because it takes years for seeing something growing because of the specific growing cycle of the orchids. They grow very slowly, and so you need to be very patient. But for the species of the dry grassland, well, actually sowing the seeds is working pretty well. I don't have any figures about that, because we are at the beginning of the process. But yeah, it's something that you need to try, actually, because usually in a, let's say, in a dry grassland where you had orchids in the past, you still have the necessary mycorrhizae. Of course, in some areas we have completely removed the current vegetation, and we have implemented sod cutting. In these circumstances, yeah, I can suspect that the mycorrhizae are away, and so, well, it's not going to be so easy to restore orchids. But for those areas where the restoration of the abiotic condition is not so profound, then you can consider seeding orchids.
Next question. What are the advantages of translocation of plants compared to sowing?
Yeah, well, I would say that I would prefer sowing if we would have enough seed. Because by sowing, from the beginning, you get the individuals that are adapted to the local conditions, because those that were not adapted, they didn't germinate, or they soon died. So you have a kind of selection. By planting individuals, you force the plants to be there, so they didn't choose that. And then you can have some mortality later on, because they don't feel very good at this specific place. The problem is that for rare species, you usually don't have the luxury to have a lot of seeds. We have, I would say, yeah, most of the time we have thousands of seeds, but it's just a few grams. And with that amount of seed, we are not going to restore on a large scale grasslands. And if we sow these few seeds, we have to keep in mind that a lot of these will not grow into adult individuals, because you lose a lot of seeds. Seeds are just food for invertebrates. They also degrade in the soil by fungi and so on. And so you lose a lot of them. And on average, you can expect that, let's say, 5 to 10% of the sown seeds can give rise to adult and flowering individuals, which is great if you have kilos of seeds. But if you don't have this situation, then we have to go through ex situ propagation just to have more. And then the transplants are functioning on the field like a source of diaspores. And like this, we can hope to get a healthy population in a few years.
It looked as if you translocate single species that first form monocultures. Or do you have experience with whole plant communities? Do you think this has benefits?
Sure. Yeah. On the pictures, it looks like monocultures, because the pictures were focusing on the translocated species. But next to these translocations of rare species, we have been implementing sowing of other species that are characteristic of the habitat to restore. And as these species were quite common, so we could work with a seed company that was harvesting these seeds in similar grasslands in a good conservation status. And then we got kilos of seeds. So next to these transplanted rare species, we also have seeding of other characteristic species of the habitat. So it's not like a monoculture. But yeah, on the picture, I was focusing on the translocated species, so it looks like that, but it's obviously not a monoculture.
Do you use symbiotic biomimetic simulations while restoring different species in the same plot?
Yeah, we saw. I'm not sure I get the point, but yeah, we saw and we planted different species in the same area. But we are not talking about plot here, because the areas to restore are sometimes very big, like 1 hectare or so. And so the translocation of rare species was just a small action within a broad grassland restoration project. So we definitely do have different species that have been restored in the area using different approaches. So here, the translocation I was talking about is just a single approach that just complements other approaches. But I'm not quite sure whether I have properly answered the question, because I was not sure I got it.
And then a last question. When sowing of seeds is better than planting, you could grow the plants for a longer time and use the seeds from them. To have more
Yes,
seeds.
that's what we are also implementing. So for the more common species, or less rare, I would say, we are also working together with a seed company who is collecting the seeds in the wild. And then they have own field where they grow the plants, just with the aim of collecting more seeds. Which is the basic work of a seed company, I would say, normal work. So this is also something that has been done in our project, but I was not talking about that because this has been illustrated in other webinars. But this is something that we also do together with the seed company.
Okay, I think we answered most of the questions. Thank you very much, Sandrine. Next week, we will have another interesting webinar given by Francisca Bruid from Sicona. She will give a presentation on the grassland restoration projects in Luxembourg. If you would like to attend other webinars of this webinar series, please register for every webinar you would like to attend. All the webinars will be recorded and can later be seen on the webpage of EXPBIO. And after this whole webinar series is accomplished, you will receive an email questionnaire on reflections and learnings. We would be very grateful if you could fill this in, as it really helps us to make recommendations for future steps in grassland restoration, so your answers are very important to us. You can still use the chat for another few minutes if you have any remaining thoughts to share, and we will check it afterwards. Also, if you think your question isn't answered totally, you can put it in the chat, and we will look at it. So, on behalf of the entire EXPBIO team, I would like to thank you all for participating, and you're welcome to the upcoming webinars. See you next time.